So welcome everyone online and on site. This is a hybrid session titled Understanding Adaptation and Evolution of Earth Organisms to Extraterrestrial Environment. I am your virtual chair, Marina Walther Antonio from Mayo Clinic. I have a co-chair, Dirk Schulze-Markuk from the Technical University of Berlin, but due to family matters, he won't be able to attend. But we do have Frank Rosenzweig from the Georgia Institute of Technology, just right there, and Flesh and Bone to help with the session for those that were physically there. We have a tight session, 6 speakers, with 10 minutes for presentation time and then 5 minutes for Q&A. So I'll go ahead and the first speaker can start the presentation, a virtual speaker, Yuguan Liu from the Mayo Clinic. Go ahead, Yuguan.
Okay, let me share my screen. Good afternoon, everyone. My name is Yuguan Liu, and today I'm presenting on behalf of our contributing authors on our recent work on non-random genetic alterations in cyanobacteria Nonstop species exposed to space conditions. So the overall motivation is to try to find out a bigger question about what genetic mutations can occur in microbes after their exposure to space and simulated Mars conditions. And this experiment is part of a larger effort called Biomax project, that is biology and Mars experiments. And the goal is to identify the genomic variation of Nonstop species exposed to Mars and space conditions. So the sample first cultured in a natural condition on Earth, and part of them were sent to the International Space Station and in flight for 15 months, and part of them remain in the experimental simulation chamber on the Earth. And after the ISS returned the sample to the ground, and they were processed and sent to us at Mayo Clinic. And we have a microfluidic and optical tweezer system that will be able to handling single cells. And what we did is to rehydrate the cells and fed them into microfluidic device and use our single optical tweezer to select and manipulate single cells into the individual chambers in the device and perform cell lysis and whole genome amplification. And then we collect the sample for downstream sequencing and data analysis. So from this work, we were able to recover almost complete genome for the Nonstop species from the single cells. And we can see some light contaminants, but they were easily removed prior to the data analysis. And we have a more comprehensive analysis on the quality of the single cell data using the single cell technology in the microfluidic device, which is published in iScience very recently. The first thing that we looked at the data set is Nonstop species sample purity and genome coverage. In general, we have observed that the samples on ISS displayed higher purity than ground samples, and especially it's interesting to see that on the ISS, even the samples that are exposed to UV, they showed 3 times purities and the ground samples that are exposed to UV, although the UV on the ground is almost twice as high as on the ISS. So this can indicate that there is some complex cosmic radiation effect in the near-Earth environment that can be detrimental and challenge the survival of the companion microbial species within the sample, but only those Nonstop species that are very resilient and have very thick cell structures can survive those extreme conditions. We also have observed that the samples in lunar and Martian medium displayed relatively poor coverage and the best sample purity were achieved in BG11 media, which is a natural medium tailored to the maximum survival of Nonstop species. And this happens both on the ISS and on the ground. And this is indicating that Nonstop can better adapt to UV radiation if it's a natural habitat. So the next, we looked at the whole genome and see if there are some variants across the genome. The first set of variants we have noticed is that the biofilm associated hemagglutinin gene, and we call it a variant if they appear in at least 2 single cells. This hemagglutinin gene related gene are responsible for mediating contacts between cells and also colony formation and biofilm maturation, and also plays a key role in protect the colonies from desiccation. So this is the first of a set of genes that we have observed there are variants, both synonymous and non-synonymous. And the second area that we're seeing lots of variants is a photosynthesis associated genes, especially photosystem protein, a PSBA gene. And you can see a lot of variants here. This gene is considered the photosystem reaction center and is found in all photosynthetic organisms, and it plays a protective role in cyanobacteria from radiation damage. So the next thing we would like to find out is to see if these the variance pattern are random or they have a certain trend. So we look at some synonymous and non-synonymous variants, and we see that they happen to have some high similarity in variant profile in the same gene across different conditions. For example, you can see here, and here. Here they look very similar. We did a gene by gene and genome-wide analysis to see what's the probability of these variants being random across different conditions. From the gene by gene analysis, the results show that the variance being random is only less than 0.1%. And from the genome-wide analysis, the probability is less than 10 to the minus 8%. So the next thing we looked at is the synonymous versus non-synonymous variants, because synonymous variants do not change the encoded protein, and they are usually regarded as silent, while non-synonymous variants have a translational impact on the encoded protein. So we counted those numbers and did the genome-wide and gene-by-gene analysis of the ratio between non-synonymous and synonymous variants. And we see that many of them are less than 1, which means that all conditions exhibited preferentially synonymous variants. Indeed, there are some of the variants are non-synonymous. So in order to find out whether there is an impact on the protein structure, we did a protein structure prediction for this investigation, and we focused primarily on a PSBA protein encoding gene because this is the area that most of the variants cluster. What we have found, there are 2 things. One is shown on the image on the left. It shows that the non-synonymous variants that are showing up in ISS sample that are in the dark, but not showing up in ISS sample that are exposed to UV conditions. And this can indicate that the combined effect of ionizing cosmic rays and also UV radiation, they may not have the same effect if each type of radiation stand alone. And the other thing we have observed is that non-synonymous variants in UV exposure in the ground, but they are not showing up in UV exposure on the ISS. And this can be because of some of the unique aspects of the ground UV, such as higher UV radiation versus the intensity on the ISS. So the conclusion is that the pattern of the variants we have seen is not random, because if the genetic variants just occur randomly, they would be scattered all over the genome, but they seem to concentrate only on the biofilm and photosynthetic loci. And the second thing is that the flight sample showed higher number of biofilm encoding variants, while the ground sample showed a higher number of photosynthetic variants. And the photosystem is also changing in the space, but just not as fast as ground. But in the flight condition, the biofilm loci seems to be a hot spot, and which can be accelerated in its evolution. So the next step it will be interesting to investigate different substrates and different species, and to see if there is a genetic variation pattern, if it's species dependent. And it would be also interesting to perform microbial single cell transcriptome and proteome analysis to investigate functionality of these variants. In our lab, we develop microfluidic tools for bacterial single cell study. For example, the lyses are very difficult to lyse bacteria, and also single cell RNA-seq, as well as the tools that can rapidly detect femtograms of bacterial DNA very quickly. So we acknowledge all our collaborators and and all our funders, both individual and as a funding agency. And thank you very much for your attention.
Thank you, Yuguan. Are there any questions either on site or virtually? And while we wait, just for quiet, there's one.
Oh, yeah, I have a question. Is this on?
Yeah.
I'm Penny Khan from University of British Columbia. I'm just wondering if you have an idea as to why a biofilm phenotype would be adaptive in a space environment.
In the space environment, so our assumption is that when under the stress or UV, the cyanobacteria was shown that they can release certain substances to protect themselves from the UV. So we have observed, we have tried to observe the phenotype change after they are returned to from the space. We didn't see any phenotypic change under the microscope, but just showing up in the gene. But this is something interesting that was further investigation.
Thank you.
Are there other questions?
There are.
Yes.
Marina, this is Frank Rosenzweig at Georgia Tech. Thank you for a lovely talk. I have a couple of questions about the procedures, and that is, how many generations were these experiments? And given what you know about the mutation rate in the organism and the number of generations and the population size, are the numbers of mutations that you're recovering, consistent with, the arithmetic there? So that's kind of one question. And the other question is, did you check, the fitness of these phenotypes with respect to the ancestral strain that was used to found these evolution experiments? Thank you.
So for the first question, these are we have we used the sample directly returned from ISS and fed into our microfluidic device to perform a direct sequencing. So what we have seen here is possibly represents the damage occurred during the process. We didn't do the generation of culture afterward.
So these cells were or were not replicating when they were on the ISS?
They're in a lyophilized condition, so there are minimal biological activity over there.
Okay, all right, got it. Thank you so much.
Yeah.
And how about checking the fitness of these phenotypes that developed?
Right now, we use this technology to kind of give a proof of concept that this is something we can do. We didn't go deeper into that discussion in this work.
Thank you.
Thank you. So in interest of time, let's move on to the next speaker, which I believe is on site. Dikshant Gautam from Northern Illinois University. There we go. Very good. Thank you. Go ahead.
Well, that's All right. All right. Can you hear me? Yeah. Okay. My name is Disant. I'm from Northern University. I'm doing my PhD there. For the past few years, major part of my research has been looking at this pigment that is responsible for giving the green characteristic green color to our planet Earth. As a astrobiologist, we know the importance of chlorophyll for the biosignatures. And most of the chlorophyll that we see floating around is mostly chlorophyll A. There are other rare chlorophyll like chlorophyll D, which is found in Echinochloris marina from the cyanobacteria. And we don't know, we haven't characterized the gene for it. But that chlorophyll D is very important for us because it absorbs light around the like the near infrared region of the visible spectrum. And it is important because most of the cooler star emits light around this wavelength. It is important up there for like up there in the space for exoplanets habitability, and it is equally important down here on the Earth for if you want to grow crops that can grow in a area where there is lower amount of sunlight. Then if we can characterize this gene and transfer them to the plants, then there is a good possibility for increasing the agriculture production. The thing is, we know all about chlorophyll A, the gene that synthesizes it, chlorophyll B, for chlorophyll F, which was discovered later than chlorophyll D, we know the gene that synthesizes it, but we don't know the gene that synthesizes chlorophyll D. My advisor had tried to synthesize it, try to characterize the gene for it during his thesis. I've been trying to do it, so it's a long problem. So when we try to characterize or when we try to find the chlorophyll genes, we go the straightforward approach and try to look for the chlorophyll binding domains. And the way we do it is normally is that we use the sequence to divide the chlorophyll structures, and we use tools like annotation tool that can easily find the structures using models like hidden Markov models. But does this work always? Most of the time it does. But there are, in every genome annotation, you always find proteins that are labeled as hypothetical proteins, right? So what if the chlorophyll D gene is one of the is labeled as a hypothetical protein, so we are not finding it. The way HMM profiling normally works is that if you look at the lower figure, we see the matches M1, M2, M3. So those matches are calculated based on the consistent sequence that HMM finds out between the similar proteins. Along with the matches, it also takes into account the insertions and deletions of the bases. So taking the analogy of the duck, if we are trying to find the similarity between the ducks, it looks for the beaks and the legs, which are similar. The insertion and the deletion, in the case of duck, will be like the difference in the color of the head of the duck, that is the green color. So HMM trains itself with such features. In that way, it can predict other ducks, right? But what if there are some weird type of duck? HMM is failing in characterizing such ducks. So what we thought of was like, okay, now let's focus on more subtle features than these general features, which HMM model focuses. So let's go even subtler and deeper and look at the RGB values using the machine learning models. If we do that, we think we can find an even better relationship between those proteins. So, and one thing that machine learning model has is, or we believe it gives an edge over HMM model, is that along with the positive data set, like similar ducks, it also looks at the negative data sets, a feature that other species like rats and falcon might not have with the duck set. So that might give it an edge over HMM model. So we use the random forest for our machine learning model. And random forest is a collection of decision trees which can classify one category over other. And switching from the example of duck, let's go to a jar with a collection of leaves and a Bigfoot. And when we have already trained our model with positive and negative training data set, when we take out one of the objects from the jar and feed it to the model, the way the model works is that each decision tree is going to decide whether that object is a leaf or a Bigfoot. And they might have different decision, but in the end, what the majority of tree decides, that's going to be our final object. So in this case, it's the leaf. Another thing is the feature selection. Like in the case of duck, we were looking at their bills and legs. Here, for protein sequences, we are looking at its amino acids. However, the model, machine learning model, it requires features in the terms of numbers. So we use conjoint triads to extract features from our amino acid. The way it works is that it divides our amino acids into 7 classes, and it takes a group of 3. So every time it's taking group of 3, meaning 3 into, so 7 into 7 into 7 equals to 343. So we have 343 columns, and we are looking at the frequency of those 343, each of the 343 columns in the sequences, right? And how we are working on it is that it starts with the first sequences, that's shown in the figure, and it's going to classify them, like the LCS is being classified into G2, G7, and G3 class. Then it counts its frequency. Then it is going to roll over to CSS, and so on. So in this way, we classify the frequency of our amino acids. And for training our model, we use chlorophyll binding proteins from UniProt datasets as a positive training, and E. coli proteins for our negative training data. What we found from our model was that we identified around 64 genes from the model. Some of them were the photosystem genes, but the model didn't predict any biosynthetic gene. We tried to validate these genes to the Amorina core and noted that these genes are not exclusive to the Amorina cyanobacteria. These are also present in other cyanobacteria. So what does this mean? One thing it might mean is that our model might need some tuning, and we might have to use other negative training data set, even a closer one, the closer cyanobacteria. Or it also could mean that the gene is present in all the cyanobacteria. The only difference is that in some, the foot is off the gas pedal, and in our Amorina, the foot is on the gas pedal. So it's being, the transcription is there. So for looking at those, the expression of those, whether it's the expression that's really the difference, what I did was I looked at I took out the samples from NCBI that had done like different projects have been done for Amorina, looking at the different conditions. Some were done for the oxygen, some were done with the iron. And what I did was I merged them all into a single, into a single training data and testing data. I combined all the features, and I supplied it to the model, thinking that the chlorophyll binding genes will cluster into together, and the rest of the genes will be clustered far. But what I found was that they didn't cluster together. The blue are the chlorophyll genes involved in chlorophyll biosynthesis, and the yellow are the rest of the genes. So one thing that might have gone wrong is that there are lots of noises. There are noises here. When I played around with features, I found the clustering was a little bit improved, but it didn't. So the clustering here is done using a TSNE plot. So it's a dimensionality reduction plot, just like PCA. And it also does the unsupervised learning, right? So when I use this for clustering, I found, well, it wasn't clustering well. And when I tried to play around with some of the features, I found that, well, there are noises there. And if I could reduce the noise, then these genes will cluster together well. So this brings us to our future work, which is that we need more transcriptomics experiments done. So if you can add more features into in my training data, then our model will be better at clustering all those genes responsive for chlorophyll biosynthesis. We need more genome sequencing of the non-chlorophyll D groups, and using the competitive gene analysis, we can reduce the number of core genes to Amorina. And lastly, we're also planning to use deep neural network, because we have seen in many instances that deep neural network works better in identifying certain patterns. Like in the case in some experiments, they have used it to classify biofilm proteins, which were previously thought to be hypothetical, has been classified as biofilm. So, and one reason we wanted to use it for RNA-seq value is that because when we reach to the level of RNA-seq expression, we are losing, we have already processing the data using different sort of normalization. So what if we just feed in the raw data to the model, maybe the machine can find some patterns that might have been lost during this processing. So these are all our future work that we are currently doing. So in this way, I want to acknowledge the NASA's Exobiology program for the funding, GRED for sequencing, and NI for giving me the opportunity to come here and present. Thank you, everyone, for listening.
Thank you. Thank you, Dishant. Any questions virtually or on site? I just have a question while we wait. So you said that the model predicted for the system genes well, but really wasn't very good at predicting biosynthesis genes. Is this like a database issue, or what do you think is the reason for that particular observation?
I think the model must look didn't get the features that are very exclusive to cyanobacteria. Right now, I modeled it with E. coli, so maybe it was able to differentiate between E. coli and cyanobacteria, but it didn't find a model that is very exclusive to, or features that is very exclusive to cyanobacteria. So I think it will work better if I add in some cyanobacteria also for the negative training data sets and train it so that it can easily recognize the features of cyanobacteria, so that I believe it's going to work well on that.
Yeah, it looks like you can reach out to U1, has all the Nonstop and cyanobacteria. Might be a way to collaborate. Very good. Any other questions from anyone?
We have one in the chat.
Yeah, please go ahead.
From Diana Gentry at NASA Ames. Did you have a toolkit in mind for your deep neural network future work?
Sorry, I didn't hear it well. Something on neural network? What was
The
that?
question is, did you have a toolkit in mind for your deep neural network future work?
A toolkit? Did you say toolkit? Sorry.
Toolkit.
I'm still planning. I don't know. There are different like convolution neural network and other things that people have been using. So I need to explore more on that. So right now I'm just exploring. I'm sorry, does that answer your question?
Probably. Are there any follow-up questions? So you're going to explore the transcriptome, you said. Are you also going to explore more the proteome functionality of prediction models, or just more genome and transcriptome?
For the raw sequences, it's also gonna be the prediction model. It's gonna predict the 0 and 1, like whether it's chlorophyll binding and non-binding. But yeah, the features that I'm going to supply might be different. Like I might extract features differently.
Pretty good. Yeah, I think you and you need to talk because you can help with functionality for her data set, and she can help you with the database for the cyanobacteria. So this is great. Any other comments or questions from anyone? Otherwise, I think we're about on time. So thank you, Dishant. Great presentation. Appreciate it. And we can move on to virtual speaker, Joseph Stone. Are you ready to go? Awesome.
I am.
Joseph Stone from Brigham Young University. So please go ahead.
All right, so today I'll be presenting on the microbial metabolism measurement system, which is a device to simulate the impact of habitability stressors on microbial cultures. And I'm presenting on behalf of the NASA Ames Research Center. So the system, I'll just call it the system for brevity in this presentation. There's potential extraterrestrial habitats that can be limited by a number of different stressors, including ultraviolet radiation, ionizing radiation, high and low temperature, lack of water and other nutrients, as well as high and low pH. Each of these stressors will individually limit microbial growth and survival, but when they're combined, they have additional effects. And so these combined stressors are key to our understanding of how Earth-like organisms can adapt to harsher environments that are found outside of Earth. And our system is, its purpose is to measure the effect of combined stressors in real time autonomously. So The system is an automated liquid culture system equipped with environmental control and multiple embedded sensors. You can see in the images below, on the left is the vessel which controls which holds the culture, as well as the sensors. And then there's also an additional sensor that has recently been added, the optical density sensor, the communication circuit, which controls the sensors, and then the environmental control on the right is a UV LED housing. And then to go into more detail on the sensors, the oxidation reduction potential is able to measure consumed or available metabolic energy. The electrical conductivity and potential of hydrogen sensors can measure osmotic stress or metabolite production. The dissolved oxygen measures anaerobic environment or oxygen consumption. And the optical density sensor can measure the population density, as well as some temperature sensors to monitor the conditions and cannula that can inject into the medium or headspace. Each of these sensors can be used with the environmental control to control the environment, or just to see how the culture is doing and the response it has. So one example application of our system, BioSentinel, is a cube satellite, which is the first to go beyond low Earth orbit with a biological CubeSat. They have yeast, and they're going to be determining the effects of space radiation on this yeast as a model organism for future space missions outside of low Earth orbit. And so what our system is doing is it's on the ground as a part of some ground experiments. What it does is it's able to take more detailed measurements of the same assay than BioSentinel, which will allow the researchers to interpret the data and be able to understand what is going on with the cells. It's also has a history of past biofluidic satellites that have used a similar assay, and it will be useful for future projects. So recently, some changes were made to this system. There were some problems with it being partially reusable, not airtight, not as robust, as well as too much headspace. And so some improvements were made. You can see a reduction in the headspace, as well as airtightness was achieved with gasket, threaded adapters for cannula and probes, as well as clamps. And the plastic threads were replaced with metal, and rubber parts with autoclavable material. Another part of the system, there was a previous version of the optical density sensor, which used pneumatic force from an air cylinder to drop the media and then determine the population with LEDs shining through it. The problem here was there's a deformation of the silicone on the plunger, and it wasn't able to drive. As well as you can see, it's in not a temporary form. And so some improvements were made to the system. A printed circuit board was designed, as well as the actuator was replaced with one that connected directly to the plunger in order to reliably drop the media, and it improved optical resolution and signal integrity. Here's a video showing the prototype for the new sensor. It draws up the media, and then the LEDs shine through to determine the density. And all of this is being controlled by a Raspberry Pi computer, which is more integrable into the current system of the other sensors. The last part of the system, the communication circuit, which controlled the sensors and fed the information to the Raspberry Pi computer. Again, it was in a breadboarded format, and so it was prone to disconnect, occupies a lot of space, and susceptible to water damage. And so, again, improvements were made to by designing a printed circuit board, which made it more immune to movement. Having soldered parts, the copper tracks in the circuit board allow for tighter connections, reduced the size, and made it able to be mass produced, as well as inherent to a printed circuit board, improved the signal and power integrity, as well as water resistance. And so why is this system important? You can see that on the left here, well, there's a number of different applications this system can be in for different microbes as well as different forms of experiments. On the left here are just some charts showing another example of E. coli being exposed to ultraviolet radiation across a number of iterations. And in the bottom, you can see all of the sensors inside the system showing the response that it has over time. On the right is another example application of a slightly different form of the system, which is being used as an analog for Europa waters. And you can see in the bottom the different sensors connections there, as well as ports for the gas and the headspace venting. Here, so previously I talked about how it's able to have real-time autonomous data. Here's a video showing collection of data. On the left beaker is a system or a culture that just has, that doesn't have a metabolic indicator dye, and on the right it does. And you can see that on the right, the graph is slightly different, showing that there are different growth parameters associated with the metabolic indicator dye. And so again, this system, because of its ability to integrate multiple sensors into one form and control it with software, it's able to enable researchers to have a system that's able to measure this in real time, as well as multiple parameters, which makes it easier to perform the experiments. And I'd just like to acknowledge Tao Sheng, a fellow intern, for his feedback on the presentation, as well as Chimai Govindaraj and Chloe Lacates for the Europa application information, as well as my internship coordinators, Portia Parker and Haley Feck. And thank you. I'll turn the time now to questions.
Thank you, Joseph. Great to work on time. Any questions from anybody, virtual or in person?
We do have a question from the
I'll
chat.
ask one, Marina, and that is, can this be adapted for continuous culture? That is to say, to do evolution experiments, or is this for relatively short-term physiological experiments? What do you guys envision?
Yes. Yeah, it can be adapted to that form. A previous form or another form is actually using fluidics cards, and it's able to, just with the fluid flow, I showed on this slide, this was exposing the E. coli to multiple different iterations. So yeah, it has been used before. A different form of this system has been used for experimental evolution. And so, yes, it can easily be adapted to that.
Great, thank you.
I think I heard that there was some other question maybe online, was it?
Yeah, we have a question from Laura. It says, hi, Joseph, very cool. Have you considered accounting for the impact of microgravity, for example, by integrating your setup with a random positioning machine?
I have not considered that, but that's definitely something that we can look at. This right now, this is mainly used for ground experiments, and so we haven't really thought of I haven't thought of other applications, but that's a good thought. Thank you.
Other questions? So I was thinking as I was watching you, you had the two chambers, for example, you were comparing with different sensor. And I was thinking, could you utilize this, say, for more of a life detection mission sort of thing to determine if a sample is, has some kind of biology in it, biological agent, by just having basically replicates of the sample, one sterilized and one not, and see whether that changes those markers they're measuring. I mean, in this case, temperature, pH, and other things, but you could measure whatever you like, and just to see as a difference, could you utilize it for that sort of thing?
Yes, I think you could. Yeah, you'd probably you'd have to adjust the form of the vessel culture, obviously, but these kinds of sensors, I think, would be able to detect that. And with the BioSentinel mission, for example, using a metabolic indicator dye, it's actually improving that assay by giving more in-depth look at how that indicator dye is parameterized by all these different, the pH, the electrical conductivity, and so gives a wider picture. And so, yeah, I think that with these parameters, you can adapt it to that kind of system.
And even through time, right? You could track whether whatever is there is growing and or reaches an
Right.
equilibrium of sorts, right? It's pretty very interesting, actually.
Yeah,
Great.
yeah. There's a lot of
Any-
applications for this system.
Yeah, there are. I'd be interested. A system like this for purposes too. This is very neat. Other questions from anybody else?
Yes, can you hear me?
Yes.
Hi, Rosa Santomartino from the University of Edinburgh. Thanks for this work, which was very great. And I was wondering, do you think it would be possible to add substrates like regolith, for instance, or is it going to damage the sensors?
Add what? Sorry.
Regolith.
If you can add regolith or other substrates as well.
I'm not sure what kind of damage would you be worried about with that?
Like, for instance, you could study, well, I work on biomining, so I'm always thinking about that, like interaction of the microbes with the regolith, maybe some Martian analogs or lunar analogs, or even just terrestrial rocks, something like that. I don't know if it's going to damage the sensor. That's why I'm asking.
Yeah, so I'm not sure on that. I could get you in contact with my mentor, Diana Gentry, who has a little bit more understanding of the mechanical impacts on the sensor, but I'm not sure. I don't believe there would be damages to it.
Okay.
Yeah, I think
Thank
what
you.
Rosa is asking is in terms of, even physical, because there's movement, right, in your system. There's some motion that kind of keeps mixing, right? So if you
Right.
have kind of, abrasive substrates that could cause physical damage to it, or corrosion as well, right? That could also damage some of the components. I think that's kind of where the question is.
Yes, and it could also cover some sensors, I guess.
Or stick to it, or right, kinds of things, right?
Yeah, the system right now is pretty robust to any corrosion, but I'm not sure for all different kinds of substances what the impact could be.
Okay. Thank you very much.
Thank you for your question.
Any other questions? Otherwise, I think we're good on time. So thank you, Joseph. Much appreciated. And we can move on to Eric Wingarten. I believe he is on site from the U.S. Army Engineer Research and Development Center. Go ahead, Eric.
Okay. I'm already messing it up. No. Okay, so this is me. Give a computational biologist the computer and all right, we got it figured out. Okay, so yes, I'm Eric Weingarten. Glad we've got enough room here, everybody. So I'm going to be talking about microbial communities in salt pan sediments and their results in response to a simulated Martian environment microcosm. And these are sort of sourced from an unusual terrestrial analog. And I'm going to go through sort of some of the benefits of using this type of system. So salt pans, also referred to as salt flats, are coastal ecosystems. So you hear a lot in terms of analog environments. You know, people go to lots of very far-flung places. Antarctica, the far Arctic, the Atacama. These are all very exciting places. I went to the coast of Mississippi. I'd like to think that it's exciting in its own ways. But this system has an interesting hydrology. In addition to being hypersaline, so some of these salt pan ecosystems, salt flats, especially ones that exist in China, can be up to 20% sodium chloride, so 200 parts per thousand. The ones that we are working in are slightly less saline, but still certainly hypersaline. And they go through these phases of matrix osmotic stress, where they go through long periods of desiccation and very short periods of hydration. And so they serve as a very good analog for just two parameters that we suspect will come into play in Martian RSL or potentially subsurface brines, where we're looking at hypersalinity and matrix osmotic stress. And so the way that these salt pans form is that they are sort of inland coastal systems. So normally they receive no hydration, but they become hydrated during storm tides, so during hurricanes. And so you can see in the diagram here, normally these are completely unhydrated. When storm systems come through, the particular system that we were looking at was before and after a tropical storm. Water pushes in, and because these are inland systems, the water just cannot retreat back out with the tide. And so you sort of get these small tide pools that exist inland. But then what happens is this water evaporates away, and all of the solutes in that water precipitate out. And as this happens over geologic timescales, you build these deep salt crusts. And you can see the little diagram here. You sort of have these sand flats that exist on the perimeter, and then you get these small brine ecosystems that exist for long periods of time with very high salt concentrations right in the middle. And why these are potentially good models for Martian soil is that they have high salinity. So like I said, the sites that we are looking at only gets up to about 50 parts per thousand. Other salt pan ecosystems can spike up to 200. But they also experience high UV. So these are unvegetated systems. This is our site in the desiccated at the top and hydrated at the bottom states. And so you can see that at these salinities, you're not going to get any coastal vegetation that's going to form. And because they're unvegetated, they also experience locally very high UV. And they're also going to be dealing with this hydrologic condition where you're going to have periods of prolonged desiccation and low water availability. And so when we're talking about Martian RSLs, from the microbes perspective, you've sort of got to balance the potential damage from either osmotic stress from things like sodium chloride, or particularly in Martian brines, we're looking at perchlorates, which are going to have chaotropic and other toxic effects. And so you sort of have to balance how beneficial is the salt presence for retention of liquid water versus the pressure that's put in by hypersalinity. And so essentially in the desiccated state, we're thinking about the Martian equator in the desiccated state, and then in the hydrated state, we're thinking about these RSLs when they are potentially flowing. I know the origin of RSLs is contentious, but this is assuming a hydrologic source for RSLs. And so just some quick assumption testing. So this was, a lot of the research that this stemmed off of is actually global change biology research. So this is in association with other coastal research studies, but right off the bat, these are three wetland types: fresh marsh, brackish marsh, and salt pan. And so you can see right off the bat, on the left is the bacterial community, on the right is the archaeal community. And so you can see they are distinct, which is a good thing. And then separating the surface samples from the subsurface samples, which we would assume, just based on oxygen availability, that there's going to be strong differences in these compacted crusts between the surface and the subsurface. And basically this indicator species analysis is showing that the surface community is dominated by a rich abundance of extremophiles. magnesium perchlorate, sodium perchlorate, or all salts combined. So for a total of 24 different treatment levels. And then they were incubated in anaerobic jars that were, the oxygen was removed, a CO2 atmosphere was generated, and they were incubated for 21 days each at -20, +27, and -80 degrees in series, which is a relatively good approximation of Martian equatorial average high and low temperatures based on rover readings, with a little bit of artistic license because any laboratory biologists know that those are pretty easy numbers to model in the laboratory. And then 16S, ribosomal RNA for tracking compositional change, metatranscriptomics, as well as culturing onto saline media to track survivalship. And so what we see, this is our survivorship table. So colony forming units on the y-axis, experimental days on the x. And so you can see that each perchlorate treatment had relatively similar survivorship, no significant differences. So each of the 3 perchlorates that we tested had similar results. And they were all significantly lower than the no salt group. So the one that we did not amend with either chloride or perchlorate actually survived these conditions better. Overall survivorship diminished over time significantly, but there was no significant change within any single treatment. So overall, we're seeing that these communities were able to survive this analog experiment, but the perchlorate treatment groups had a lower survivorship than the ones that we did not amend with any salinity, and I'll get to potential implications of that in a second. Another interesting finding was that the fate of the microcosm communities was strongly tied to the source sediment used, whether that was the dry surface, dry subsurface, or wet subsurface. The 2 ordinations on the left, so Bray, Curtis, and Jacquard, that's looking at changes in community abundance and community occurrence. And you can see that they're sort of, the big squares are starting conditions, and you can see that we sort of shotgun out into separate corners. And so in terms of the bacteria and archaea that are present, in terms of their relative numbers, we have different responses from these different environments, but Unifrac is an ordination method that is looking at taxonomy, and you can see that we get a shift to the left across all treatments. And what we hypothesize is that that is essentially we are selecting for nothing but extremophiles. And so different environments were better or less adapted to the environment, but the things that survived across the board were all of these extremophiles. And then there's a lot of ordinations here, but essentially the 2 left columns versus the 2 right columns, we see that the surface community had a relatively consistent response, whereas the subsurface community had a shotgun response. So there was no consistent response in the subsurface. In the surface, we had a similar response across the board, across all metrics. And we hypothesized that that's because at this surface, where we have higher salinity, higher UV, and higher hydration variability, you're getting a similar selection for nothing but extremophiles. And so they were better adapted to this environment that we're modeling. And then finally, looking at the response here, a great big heat map, but in summary, the dominating surviving lineages were dominated by halophiles, which I've highlighted, thermophiles, psychrophiles, and radiation-tolerant organisms. So you can see roughly half here are known lineages that are extremophilic and extremophilic for the conditions that we were testing for. So in conclusion, we demonstrated the good news. We demonstrated sustained viability of a mixed natural archaeal and bacterial community to Mars analog conditions for 63 days. And we observed a shift in taxonomy towards known extremophiles that would be at least hypothetically, well adapted to Martian conditions. So the key methodological finding is that there is some benefit to doing things through a whole microbiome approach rather than a stock culture approach. The bad news was that growth and sequence counts declined significantly when exposed to chloride and perchlorate. So in terms of thinking about chlorides and perchlorates in terms of their eutectic or benefits for drawing liquid out of the atmosphere, deliquescence, that's the word. In terms of those benefits, that provided relatively little benefit relative to the added osmotic and chaotropic toxicity. So this is where I'm talking about when we're starting to think about these Martian brine ecosystems and their habitability, it shouldn't be taken for granted that hypersalinity is a good thing because it's going to provide liquid water. The conditions have to be right that the temperature and liquid availability needs to be right in that Goldilocks zone for the trade-offs in terms of toxicity to be a benefit. And so we show that mixed analog soil communities can serve as agnostic predictors for adaptation to extraterrestrial environments. So I would encourage others to explore that approach as well. Thank you to my co-authors, especially the field personnel who helped identify and collect these samples. The results are going to be in Astrobiology in the July edition, I think, and NASA for funding, and I'll take any questions.
Thank you, Eric. Any questions, online or in person? And I'll go ahead with one. So you picked up those samples and put them in, -20 or -80 in different conditions, chloride and perchloride. Is there interest in trying to do that in a step-by-step manner? So kind of allowing acclimation of the samples through time to kind of adapt to it slowly rather than a kind of abrupt change that could improve a lot of the, growth rates and all that. Is there interest in pursuing something like that?
Yeah, I would ask, can I put that question in writing on my current grant proposal? Because I work, interesting employer for this meeting. I work for the U.S. Army. I'm currently applying for DARPA funding because there's lots of DARPA interest in basically building synthetic biological communities off of various exotic soils. And so what we're planning to do is sort of take this was sort of a ham-fisted example of directed evolution. We would like to be a little bit more systematic with it and basically piecemeal out different levels of these solutes into different soils and grow them over a series of generations. And really what we're planning is, in addition to sort of that forward directed evolution, over time we also want to take out subsamples of both the soil media that we're generating and the communities that grow off of them. And then we can backwards challenge and basically, generation 10, can we put generation 10 back into a generation 2 soil, and are they now better adapted to those conditions? So that's what we're planning.
It's great, and I think you can even use Joseph's system.
Yes, yeah, I was thinking that too.
Very cool. Any other questions? All right, otherwise we're up on time. Thank you so much. Really, really very, very interesting presentation. And I want to see that follow-up work, really very interesting. All right, we can move on to the next speaker, Rosa Santomartino from the University of Edinburgh. She's on site, I already saw her, so go ahead.
Next presentation, I guess. All right, so thanks for being here. Today, I'm going to talk about what we learned from 2 space microbiology experiments that we performed in 2019 and 2020-2021, BioRock and BioAsteroid. And what we learned from that from the perspective of microbial interaction with terrestrial and extraterrestrial rocks. So microorganisms perform a variety of different tasks, and for us on Earth, I'm sure I don't have to convince you about that, but today I want to focus on one in particular, which is the process of biomining. So what is that? It's the process of using microorganisms to extract metal of economic interest from rock ores and mine waste. It is also used in other similar chemical reactions are also used in some other processes like bioremediation, for instance. Both chemolithotrophic and organotrophic microbes can be used for this. And on Earth, it is currently used to extract a variety of different metals like copper, gold, and others. There are, in general, some advantages of using biomining in respect to traditional mining because it is generally considered complementary as well as more economic and environmentally friendly. Now, because of how many tasks microorganisms perform on Earth, imagine how many of them could be actually translated beyond Earth, for instance, on an extraterrestrial settlement. And some of them include the generation of food and oxygen, the recycling of waste, the production of fertile soil out of extraterrestrial regolith, and also in some space bioindustries. Now, you may notice that all these possible roles for microbes beyond Earth have all the final aim to sustain life support systems. And this is also related to the concept of in situ resource utilization, which is just the concept of utilizing the resources in the place in which we are going to settle rather than relying on a constant resupply of resources from Earth, which the farther we will go from Earth, the less become sustainable, of course. And from this perspective, the possibility of performing biomining in space is rising and increasing interest, and maybe it's easy to understand why. Of course, the possibility of using microbes to extract elements, useful components, materials, in the place in which we are going to settle is quite intriguing. From this perspective, the planetary bodies of interest are Moon, Mars, and asteroids. Moon and Mars because they are regarding like space agencies think that these are the future of human space exploration, and asteroids because they contain some key and precious elements. However, the conditions that are present in these planetary bodies are, of course, different than those on Earth. And just to mention one, gravity is different. It is lower in respect to Earth in general. So if we want to understand if we can perform biomining in space, we first need to understand how gravity conditions that are present in these planetary bodies, for instance, gravity, influence the way in which minerals and microbes interact with each other. We cannot assume that what happens on Earth is going to work even in space. So with this question in mind, we performed the first two biomining experiments on board the International Space Station, BioRock in 2019 and BioAsteroid in 2020-2021. So let's go for BioRock first. BioRock, as I said, was launched in 2019, but it was first proposed by Professor Coquelle from the University of Edinburgh in 2008. And as you can see here, a variety of different groups made it possible. The first thing that we needed to do was to select the right microorganisms which needed to have some characteristics in common among them. Eventually, we decided to go for these three microorganisms here. They are all bacterial species: Sphingomonas dissecabilis, Bacillus subtilis, and Cupriavidus metallidurans, each one provided by a different group. But we also needed to select the right terrestrial rock substrate, and we selected basalt mainly for two reasons. The first is that basalt is quite porous, so it provided a good support for bacterial colonization, and the second is that it is widely present on both Moon and Mars, so it provided basically a good analog. So we've selected one specimen from Iceland. We cut it into basalt slide. You can see here on the right, maybe I have the arrow, yes. You can see here a basalt slide, an example of that, which was roughly 15 millimeter wide by side and 2-3 millimeters thick. Then we inoculated every basalt slide with a single bacterial culture, but we also had some non-biological control, which was just some sterile basalt slide. And we have put them into the biomining reactor, which was an hardware that was specifically designed for this experiment by our group and Kaiser Italia. And then it was launched on the International Space Station in July 2019 on SpaceX Commercial Resupply Service 18. Here you can see, well, here this is me very happy on the launch site. And this is the astronaut Luca Parmitano installing the samples on board the International Space Station. When in space, samples were subjected to either microgravity, simulated Martian gravity, or simulated terrestrial gravity. But we also had another set of samples, identical to the others, which was, we call it ground control. It stayed on the ground, so on Earth, and it was subjected, of course, just to true terrestrial gravity. Samples grew in liquid culture in the presence of the basalt slide for 21 days at 20 degrees. And after this period, they came back on Earth, and we analyzed them. So let's go to the results. What did we discover out of this experiment? The first thing that we tested was the final cell concentration, and we analyzed that by both direct cell counting and optical density. What you can notice from these results here is that you can find some differences between some of the ground controls and some of the space samples, which are indicated here by the asterisks. But in general, the main result here is that when we compare the space samples, so all the samples that were present in space, we found no significant difference in final cell concentration. What does it mean? From a biological perspective, or if you want a space biotechnological perspective, this means that after 21 days, the bacterial cell culture all reach the same final concentration regardless of the gravity condition. And this may be a good indication that biotechnological application happening in lower gravities in respect to Earth are not going to be negatively affected by gravity, so they may be possible. The other thing that we focused on is biomining, of course. So we focused on two main type of elements, rare earth elements and vanadium. Both have high industrial interest, and we believe that they may be important in a future space application. And what we found is that at least 1 out of the 3 microbes, 1 here and 2 in the case of vanadium, were actually able to extract these elements in all the gravity conditions that we tested. So basically, the results that I show you here, the 2 microorganisms that were able to do that were Sphingomonas dissecabilis and Bacillus subtilis. And these results indicate that space biomining is in principle achievable under a range of gravity conditions. We also tested biofilm formation on the basal surface, and as you can appreciate from this picture, while Bacillus subtilis didn't form much biofilm on the rock surface, both Sphingomonas dissecabilis and Cubriavidus metallidurans have a positive trend of biofilm formation on increasing gravity regimens, and I hope you can appreciate it from both the pictures and the fluorescence quantification on the left. Now, after BioRock, we decided to go for a second space biomining experiment called BioAsteroid, which is a little more recent. It was launched in December 2020, and our samples came back on 2021. It is quite similar to BioRock, but there are some key differences. For instance, in the microorganisms that we used. This time we use the same bacterium, one of the 3 bacterium that we used for BioRock, which is Sphingomonas dissecabilis, because it behaved quite well in BioRock. But we also decided to expand our knowledge on microbial behavior, basically, in these conditions, by adding a fungus called Penicillium simplicissimum. And we also selected a mix. We didn't select. We decided to go on for a mix of the 2. We also changed the rock substrate. In fact, instead of using terrestrial rock, as we did in BioRock, this time we decided to go for real extraterrestrial rock, to be more specific, crushed L-chondrite. The experiment, again, is quite similar, but it's a little smaller in respect to BioRock. In fact, we only had some samples launched on board the International Space Station, which were subjected to microgravity, and another set of samples that remain on Earth, and it was ground control. Apart from that, everything was quite similar, but apart from the fact that instead of having 21 days of growth, we had 19 days. So do we have any results? Because the samples, the experiment was more recent, the results are still under analysis, but I can show you something. For instance, when we analyzed the pieces of meteorite that came back, so after the space flight, under scanning electron microscopy, we saw that the bacterial species, the samples that were in contact with the bacterial culture, did have some feature indicating that biofilm formation actually formed on the meteorite rocks, and you can see that with the red arrows. But maybe more interestingly, when we checked the samples that contained the fungus, both the mixed and the single culture one, we saw mycelium formation on the top of the rock pieces. From the perspective of biomining, this time we focused on another group of elements called the platinum group elements, which are quite precious type of metals. And as you can see here, these images here show that we found evidence of fungal extraction of palladium here, which is one of PGEs, from non-meteoritic materials in space. So, to summarize and conclude, with these two experiments, we demonstrated for the first time biomining on a space station, particularly of some precious elements like rare earth elements and PGEs, from both terrestrial and extraterrestrial rocks, and using both organotrophic microorganisms. We demonstrated that biomining is in principle achievable under a range of gravity regimens, and also that the final cell numbers were not affected by the gravity condition after 21 days, which is a good indication of space bioindustrial applications. We discovered the novel biomining microorganism, which is Fingomonas, and we also demonstrated for the first time microbial interaction with meteorite material on a space station in microgravity, at least to my knowledge. But there is a lot that we still need to understand. For instance, we need to have more insights of the effect of gravity and other spaceflight conditions on the microbial processes. We need to select the best microorganisms for any given application that you want to do in space. We need to scale up the system and develop technologies of that, because our reactor was 5 ml of volume, so it's quite a small volume. If you scale up the system, are you still going to have the same results? We don't know, so it's something that needs to be tested. And then last things that I want to point here is how can we use microbial biotechnology to support sustainable space exploration? And this is a project that I am starting now. It's funded by the Leverium Trust, and I'm going to focus particularly on plastic biodegrading microorganisms. So if you want to know more, just contact me or come to me later. And with that, I would like to thank both the BioRock and the BioAsteroid teams, and you all for your attention, and I will be happy to take any question.
Thank you, Rosa. Any questions online or in person? And I have one. Oh yeah, go ahead. Go ahead.
We have one.
Hi, Adam Ellington from University of Florida. First of all, amazing. I love this. Is a very exciting one.
Thank you.
So the organisms that you used were not genetically modified in any way, right? They're just the wild type organisms. And do you have any plans to try to optimize the efficiency of the extraction process or any of that through any genetic modifications?
Yeah, thanks for the question. So we, after BioRock, basically, one of the microorganisms that behaved better is Fingomonas dissecabilis. And one PhD student is now studying this. So he's studying, first of all, the mechanisms, because we don't know how it works. Actually, we didn't even know that he would be able to biomine, because we had indication that he could do that, but we weren't sure about that. And he's analyzing, first of all, the mechanisms. Being an heterotrophic microorganisms, it must be something related to organic acids, basically. And then the idea is to understand, once we understood which are the key genes, to try and modify it to see if we can improve the process.
Very good. And I think, in the interest of time, we'll have to move on. But I just want to say that maybe, speaking with you one might be interesting, because she had the findings about the biofilm preferential mutation. And, I don't think you have genomic data, and she doesn't have functional data, but it seems like there's something interesting there to talk about in terms of potentially gravity or lack of gravity interfering with biofilm formation.
Sure, drop me an email, of course. Thank you very much.
That was great. Thank you. All right. So Christian Lawrence will be our last speaker, and he's virtually here with us, and he's in the University of Florence. So Christian, please go ahead.
Yeah, can you see my screen? Yeah.
Yep, I see it very well. Thank you. Go ahead.
Okay. Thanks. I'm Christian Lawrence, a master student from University of Florence, Italy, and today I'm going to talk about the survival of a lichen species, Xanthoria paritina, in simulated space condition. And let's start with an introduction on why we choose as biological sample this particular lichen and why this lichen species. So lichens are considered poikilohydric organisms. That means that they are considered photosynthetically active when there is water availability in the environment around them. and they are considered deactivated but not dead when there is no water surrounding them. So this feature allowed them to colonize a lot of different environments on Earth, and in particular the most extreme ones. And the reasons why Xanthoria paretina refer to the physiology of this lichen, because it's able to produce the anthraquinone parietin that has UV shielding properties. And the second reason is referred to the background, the astrobiological background. Indeed, Xanthoria elegans, that is a really close phylogenetically speaking species to Xanthoria paretina, showed some of the best recovery values after treatment in ground-based experiments, but also in real space experiment like on the EXPOSE module on the ISS or the Photon M series of satellites. So the aims of this project were to obtain for the first time the Xanthoria paretina FTIR reflectance spectrum in not irradiated conditions and to monitor it in situ during treatments to evaluate eventual spectral bands changes, and in the end to verify Xanthoria paretina recovery capacity after treatments. So here you can appreciate the simulated condition we applied with a UV lamp with a sunlight emission spectrum, and we irradiated the samples in two different atmospheric conditions that were UVN2, so UV radiation under nitrogen flux, and UV back, UV radiation in high vacuum conditions. And here you can see our experimental design from the sample collection and sample preparation, and most important, the pre-exposure measures of the photoefficiency parameters that we use to assess lichen vitality, that were NVI and fluorescence variation, and then the exposure phase with 6 replicates for each treatment that were control UVN2 and UVVAC. And finally, the post-exposure phase with post-exposure measures of the photoefficiency parameter every 24 hours within the 72 hours from the exposure phase. So let's start with the results from the spectroscopic analysis. And here you are seeing to the not irradiated spectrum of a lichen. And in the Overton region, maybe you can recognize some of very common bands that you can see also in other biological samples. But the most relevant part is for sure the fingerprint part region that was very important for characterizing functional group that we can find in parietin, the substance with UV shielding properties, as we find in some work like the Edwards et al. From 2003 work. And then starting with the treatments with UVN2, in this slide, you can see 2 lines, 2 colors. So the first line, the black and thinner one, is the first retrieved spectrum, and the thicker and magenta one is the last one retrieved. And as you can notice here in the Overton region, there is the complete disappearance of a water band due to the extremely dehydrating condition offered by the nitrogen flux, and a general upshifting of a continuum of a spectrum that can be noticed here in the Overton region, but also in the fingerprint part region. And here in detail, there is a zoom on the water band analyzed through time in situ during the treatment, and the complete disappearance at the end of a simulation. Moving to the other treatment that was UVVAC. Also here, there is the disappearance of water band, but it's quicker than the previous treatment. And also you can notice a more relevant and important upshifting of a continuum of a spectrum that characterized almost all the spectrum, but there is something strange, it seems strange, here in the fingerprint part region where the peak intensities of bands seems to increase. So let's focus on this part, taking, for instance, these 2 bands that are in that part highlighted. And here you can see clearly how the peak intensities of these 2 bands seems to increase, but we need to consider the big picture. Usually in IR spectroscopy, the increasing of peak intensities is related to the increase of functional group abundance, but we need to consider a better proxy to evaluate this feature, that is the bands area. In fact, if you notice here in these two bands, there is a complete flattening of the two bands that is related to the higher and stronger effect of photodegradation offered by UV in vacuum condition. So just moving in other part of the spectrum, we can notice the usual pattern of uprising, upshifting of a continuum of a spectrum and decreasing of bands area. Moving to the ecophysiological analysis, so with the recovery analysis, starting with the fluorescence variation of chlorophyll A to assess the photosynthetic efficiency of a photosystem 2 of the algae layer inside the lichen. As you can see, there is a significant difference between the samples of the two treatments, UVN2 and UVVAC, even if there is a similar pattern in the drop-down fall after the exposure. And anyway, UVN2 sample seems to recover around the 90% of the beginning value in 24 hours. Instead, the UVVAC samples seems to recover less than the 50% after 72 hours. So this kind of results seems to be okay with the spectroscopic analysis results that assess that there is a higher and stronger effect of photodegradation in a UVVAC condition. But the other model, that is the NDI model, that measure the chlorophyll A reflectance to assess the photosystem 2 structural integrity, showed something interesting. Even if there is a similar kind of pattern in the recovery between the two treatments, UVN2 and UVVAC, we need to focus on the UVVAC samples. In fact, comparing this model, the NDI model, with the previous one, we can suppose that actually UVVAC samples did not suffer a real damage from photodegradation of UV radiation, because they were able to preserve, or better, to recover the structural integrity of a photosystem 2. But they showed a lag, or a time lag recovery, or a delayed in the recovery of a photosynthetic activity. So in conclusion, we can say to have obtained for the first time the Xanthoria parietina FTIR reflectance spectrum, and to have monitored it in situ, analyzing and identifying evident changes in some bands, like the water band and the fingerprint part region. And recovery analysis have highlighted that different treatments are decisive for recovery trends. In particular, with NDI analysis, we showed a complete recovery in 72 hours for both the treatments, but from fluorescence analysis, it came out that UV radiation in vacuum may cause higher damages effect, and in particular, UVVAC samples showed a delayed or time lag recovery in photosynthetic efficiency that is okay with our results from the spectroscopic analysis. And further analysis, we are now elaborating data from another experiment. We irradiated the Xanthoria parietina species in Mars simulated conditions. And our next step is to focus more on the substance, so parietin, in particular analyzing parietin photodegradation under space and Mars simulated condition with the IR spectroscopy. Thanks for your attention.
Thank you. Thank you, Christian. All right. Questions online or in person for Christian? So, oh, there's some. So you've identified that there's a higher level of damage, I guess, introduced by UV radiation vacuum conditions that may not destroy the photosystem, but at least delays its recovery, right? Is there an interpretation of why, or what's the mechanism behind that that process?
You mean the photodegradation in spectroscopic analysis? Because here I think we need to consider just the surface of a sample of a lichen, because lichen structure is really simple, but it's important to understand the capacity of lichen to survive in many different environments, and in particular extreme environments like the deserts, where there is a high level of light fluxes. And so here I think that is just a surface photodegradation of substances, maybe parietin 2 and other substances of a lichen. And for this reason, we assessed the delay of a photosynthetic activity restart after the exposure. And anyway, from the NVI model, we didn't see something really important as damage from this model that you can see also here. So I don't know if I answered your question.
Yeah, it was more trying to understand why is it that that under vacuum conditions why that happens. I guess it might be just the desiccation or right kind of on top of the radiation.
It's extremely more dehydrating than the nitrogen atmosphere, and maybe for this reason the UV radiation is more intense on the samples.
Right, and you didn't do any. For how long was the exposure?
It was exposed for 36 minutes with a 1.34 megajoule on square meter as total absorbed dose. So it was like 25 hours on the ISS compressed in 36 minutes.
Yeah, that's pretty intense. Any other questions from anyone in the audience? Yeah, I wonder if you were to even extend that period of time, if that would actually make it unrecoverable, right? At some point, it probably becomes.
Actually, we did it as like some kind of test experiment, and we proceed with the irradiation over and over and over till the sample was completely burnt. And after like 3-4 hours with these conditions, it was unrecoverable.
3-4 hours. Yeah, very cool. Well, I was wondering about that, but I guess you guys already did it. Very neat. All right, so I want to thank every speaker for keeping us on time, and really a great session, and very much synergistic. I do encourage all the speakers really to get in touch with each other, because I see a lot of potential collaborations here that you can establish. So anyways, thank you very much, and I'll see you later throughout the week and today. Thank you for participating. Bye-bye.